Good afternoon, everyone.
Good afternoon.
Okay. Okay. We'll continue with the last class. So the last class we started with the unit 9.3, correct? So population genetics we started with. So yesterday we had an issue with the rain, so I had difficulty in connecting to internet, and it was continuously disconnecting. So instead of having that difficulty, I thought it's better to indeed have a class today. I think I gave a problem homework to work on, right? To you, let's begin with that first, and then I'll continue with the remaining factors affecting Hardy-Weinberg equilibrium. Did you guys get a chance to solve that problem I gave you?
Yes, sir.
Yeah, I shall come back to your question towards the end. Just remind me that I see that you have sent me a question. Okay, we'll work on that. But let's first begin with the. I want to know whether you've found answer to that question, the homework I gave, and if you have any difficulty, I'll explain that. So the question was on the PTC, right? So PTC testing, we looked into a hypothetical population where the question was asking, it gave numbers to you, right? There are totally 1,000 individuals. Out of them, 640 are testers, 360 are non-testers. It may feel like you know everything because the numbers are given to you. You have testers the number of people who can taste PTC and number of people who cannot taste PTC. But you have to put that into the genotypes as well, correct? The question asks you to find out the number of people carrying certain amount of gene combinations or allelic combinations. So for that, you have to work on given the information. So clearly, we have to begin with the non-testers number. I was telling you briefly about that. So have you found out the answer to this? I think two components are there to this question. The first one is, what are the frequencies of the two alleles? Each allele, P and Q, for example, what is the frequency and how many testers are carriers of the recessive alleles? So to the first question, what are the frequency of two alleles? Have you calculated that? Sorry, power is gone, so you may not be able to see my image here, but screen should be visible to you. So can any of you who answered this question, how could get this question figured out? Can you volunteer to tell what answers did you get?
Sir,
0.4 and 0.6.
0.4 and 0.6, which is higher? Like it's testers and non-testers. What is 0.4 and what is 0.6? P or Q?
non-testers are higher.
Okay. 0.6 and 0.4. Okay. And 0.6 is for P or Q?
Q, recessive. Non-testers are higher.
Q is 0.6 and P is 0.4. Okay. Yash and Sunil, you got the same answer?
Sir, I
Yes,
didn't
sir.
get the same answer.
You got the same answer? No.
No, sir, I got a different.
You got a different answer. Okay, that's all right. I'll explain that. Okay. What about you, Yash?
I got the same.
Okay. Let's see how to do this problem first. And Vishal and Yash, what about the second part? So the second part of the question, I asked you to find out
Carriers.
carriers. So carriers refers to what?
Means
Heterozygous.
Heterozygous, correct? So PQ or capital
Yes,
D and
sir.
small t combination. What did you get for that?
We got
0.48.
0.48.
0.48. Is that true with you, Yash, as well?
Yes, sir.
Okay. Sunil, I doubt if you got this answer because you have to know P and Q to answer this, correct? Okay. I'll share. I have actually given the description of this in the handout. I'm sharing that with you. So look at this. Briefly go through the answer here. So what is given here? We have given that 360 out of 1,000 individuals. That is 36% of them are non-tasters. Correct? And already given in the question itself, the hint for tasters and non-tasters, because it said, so tasters are dominant type. Tasters characteristic is a dominant type. So capital T, small t are tasters, correct? So genotypes of tasters could be either capital T or capital T small t. Wherever you are unsure, do stop me. Okay. So capital T or tasters have capital T as an alleles and only small t combinations, homozygous small t, where they are non-tasters. Okay, given that there will be a confusion between homozygous and heterozygous condition, that is capital T small t, we better start this problem with non-tasters. And for non-tasters, what we are given? 360 are non-tasters. Correct? So the 360 out of 1,000. So that is 0.36. Correct? If you divide 360 by 1,000, it is 0.36. And 360 refers to what? Non-tasters. And we know that non-tasters should be small t combinations. Correct? So small t is equal to 0.36, which is nothing but the Q square as per the Hardy-Weinberg law. P refers to the dominant allele, Q refers to the recessive allele. Correct? And Q square value out of that P plus Q square or p plus q whole square in the formula that if you see, p square plus 2pq plus q square. Q square is a frequency of homozygous recessives, which is 360 out of a population of 1000. So frequency, if you calculate, this is 0.36. A probability, if you calculate it, is 0.36. So q square value is 0.36. 0.36, but q is square root of 0.36. If you want to find out, this is q square value. If you want to find out the value for q, so square root of 0.36 is 0.6. So 0.6 is the value of q. Okay, so frequencies of the alleles, if you have to calculate using this 360 out of 1000, first you calculate the number for q square, and by square rooting this value, you will get the value for q. So q refers to small t, number of small t in that population, okay, at a frequency of 0.6. Then now you have the value for q. So from the equation, can you identify what is p value? P plus q equal to 1. Correct. So q is 0.6. So what is the p value then? So you substitute that. In place of q, you put 0.6. So p plus 0.6 equal to 1. Then p equal to 1 minus 0.6. That is 0.4. This is how you identify the p value and q value. I don't think they would ask this type of questions in the exam, but by solving this problem, you will understand the importance of Hardy-Weinberg equilibrium. Do you see that here? Purely from numbers. These numbers are just measurements, just counting in a population. So you can actually count how many people in a given population are testers and non-testers. You take a sample of them. You don't have to count. You cannot count all of them. So what do? Let's say you take 100 people, 1,000 people population. Higher the number, better the prediction. Correct? So what do you do here? So you count out of 1,000 people, how many of them are testers, how many of them non-testers, which is easy, right? You just ask them to taste tissue dipped into PTC chemical, and 650 of them are testers, 360 are non-testers. But genetically, you want to calculate how many of them are homozygotes, how many of them are heterozygotes for testers. So as we know, there's only one combination possible here, that is for the recessive character, that is for non-testers. So you derive this information by following Hardy-Weinberg equilibrium, p plus q whole square. Okay, you calculated first q square value, and from q square you calculate q value by square rooting this. And once you know the q value, we can easily calculate the p value. 1 minus q equal to p, correct? So that is 0.4. So 0.6 plus 0.4 should be 1, equal to 1, p plus q equal to 1. Is that clear, Vishal? Okay. So now the next question. Next question in this is, how many testers are carriers of the recessive allele? So how many of them are in heterozygous condition? Carriers means those that don't show the problem but carry the problem or carry the gene for the problem. So small t is the condition where the problem can be seen. And capital T small t looks like testers, but they carry one recessive allele with them in heterozygous condition. So what it is asking for? Those carriers, meaning genotype capital T small t genotype. How many of them are carrying this genotype? So what equation do you use for this? If you want to identify the genotypes from our last discussion, what equation is suitable here?
Hardy-Weinberg p square.
Right.
Plus
So
plus.
what form of this? P plus q equal to 1, is it enough?
No.
No. So we have to use p square plus 2pq plus q square equal to 1. Okay. This is what you do here. So we want to find out 2pq. This is easy. We already have p value, q value. So 2 into p into q. So q is 0.6, 0.6 into 0.4. That is 0.24. So 2pq, 2 into 0.24. 2 into 0.4 into 0.6. 0.4 is the value for p and 0.6 is the value for q. So altogether, this would give rise to 0.48. Okay, same thing in the percentages. So p square, if you see, p square is the value for capital T allele. How many capital T alleles are there in the population? So this is 0.4, correct? So 0.4 into 0.4 p square. So that is 0.16. 0.16. That is 16% of the population. 16% of the population carry capital T or homozygous dominant testers. 16% of them are capital T. And 36% are non-testers. 36% are non-testers. 0.36 is a Q square value, right? So Q square refers to what? Small t. So the non-testers are 36%. So only 16% are dominant testers. 36% are non-testers. What is the remaining value out of 100? How many are we left with? 36 plus 16 are, 36 plus 16 is 52, right? So 52% is dominant testers and non-testers. Remaining 48% are carriers. This number is carrier number, right? So does it give you the proportion or the idea about the percentage of people with respect to their genotypes? The whole problem had to begin with some numbers that you could count by collecting data. With that, using Hardy-Weinberg equilibrium, you can indeed calculate all those things we learned in Mendelian genetics, unit 9.2. Homozygous dominant, heterozygous carriers, homozygous recessives. You can actually calculate those numbers in the population. Is it really useful to calculate these numbers in a population? Just literally from these numbers that you counted by collecting data from them. This can be done for any disease, provided there is a means of identifying the disease. Whether you're hemophiliac or not, tests are there, you can perform the test and you can count the numbers. From that count, you can calculate how many of them are carriers. Carriers tell us the future disease burden rate. How many people would, if they actually have consanguineous marriages or related marriages happen, what is the chance of their children getting such a genetic problem? So disease burden on a population can be calculated using this. Okay, hope this example makes it a bit clear to you the purpose or significance of the Hardy-Weinberg equilibrium.
Sir, what I did is I underrooted 360 itself.
You underrooted 360, right? 360 is the number, right? You have to convert that first into the Q square value. And we were talking this as a probability, right? 100% here, probability is equal to 1. Okay.
So we are not getting the actual numbers, but we are only getting, what is the percentage of it?
Correct. You can calculate the percentage, you can calculate the probability. Both are possible. Okay. So these are not too complex mathematics for people with mathematics background. This is simple math. But converting that into the theory of genetics is a challenge here. Okay, so in this way, this is an extension of what we studied in unit 9.2 in Mendelian genetics. There we only learned dominant allele, recessive allele. Here in population genetics, we are also adding mathematical component to that, and Hardy-Weinberg equilibrium has provided a way to calculate these numbers. So you can literally calculate the genotypes of a population. Estimate. From small number, you can estimate. From 1,000 people, you can estimate it for a population. With small change, this would hold true. That is the biggest benefit of Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium can be used to show if a population is in a genetic equilibrium with respect to a particular trait. It can help us calculating carriers. I was telling you the use of this, and it can help us understand whether it is in a genetic equilibrium, the numbers are actually maintained. If they're not maintained, one gets some idea that on the impact of the forces of evolutionary changes, how different evolutionary factors are influencing it. We'll come to know. But we don't know what factor is causing this. We will only know that environmental factors are making a change and causing the difference. Okay, so the limitation here is we will not know what factor it is unless you are looking with respect to a specific factor and try to calculate. It will not tell you. It will give a overall picture of the impact of those factors. That is a limitation of Hardy-Weinberg equilibrium, but still it provides as a means of calculating carrier frequencies and with different genotypic frequencies in a given population. Okay, right. So what other areas did we cover in the last class? Factors, I think we started with the factors in the last class, right? So I know we started with mutation, if I'm not wrong. First, we finished mutation and then we finished natural selection briefly, correct? Okay. So natural selection on mutation, I talked to you, but I want to expand more on natural selection, the different types of natural selection. So I'm going to tell you briefly about that. So we also talked about migration, right? Okay. So mutation results in the new alleles. So it adds new alleles to a given population. So that would result in the variation in gene frequencies and genotypic frequencies, right? So if they ask you how mutation influences it, you should be able to explain it. First, what is mutation? How does mutation causes change in the genetic equilibrium? So this is how you have to explain it. Those that are favorable mutations will be selected by natural selection, because mutation by itself cannot do anything. Mutation provides the change, and natural selection acts on it. And wherever there is a benefit, those varieties or genotypes are favored, so that number of those genotypes increase in a population. So you have to explain this, thereby increasing the genetic diversity of a population.
Sir, one question.
Hmm.
Where we said that mutation happens and then environment, natural selection works on it.
Correct.
But in case of some diseases, so it is the outside force which is leading to mutation, right? I mean, is it the case?
What do you mean by outside force here? Are you referring
Any
to?
disease that brings in changes of adaptability in organisms.
Okay, but are you talking about genetic diseases where the disease is a result of mutation?
So it is always from mutation to natural selection, not the other way around.
Correct. Mutation is the main source, the key source for the variation. Natural selection only acts in a way that if the mutation is beneficial, it is kept. If mutation is detrimental, it is removed from the population, because I will talk about that. It reduces the fitness of the population. So what is fitness? How is that actually affected? I'll briefly explain that little later. Today we'll discuss that. But yes, mutation is a source and natural selection acts on mutation. Okay, and mutation is one form of source. There are other possibilities, recombination, and we have other sources of changes in the population as well. Correct, but mutation is the it results in a novel change, new change. Okay, all right. Migration. How does migration affect population?
Yes, it's low.
Gene pool. So you consider a population where there's an amount of gene pool there. Okay, so what happens here? If this is a population, initial population, either people coming in and going out would impact it. People coming in would bring new genes. People going away will take out those alleles. The number of the frequency will decrease. People are migrating would carry the gene pool somewhere else. And with respect to this population, you are losing those numbers. You are losing people with those characteristics. So the number reduces. Correct? You see the change in the frequency. The frequency changes because the number of those people carrying those alleles are gone. They went away from that population. Similarly, immigration when happen, people immigrate, come in. What happens? They bring in new alleles. If they're coming from a different population, they bring in new alleles. New alleles are added to the population. That also disturbs the genetic equilibrium. Correct? So we briefly talked about this theory called one island theory. So where we talked about if the huge population versus a small island, small population, where will be the large impact? Large impact will be where people move from this continent to small one. Why? Because small one will have large impact because numbers are small here. Let's say there are 5,000 people, 1,000 people are living there. Small addition would also make a difference. Okay, so migration will have different impact on the Hardy-Weinberg equilibrium. But the thing that you notice here compared to the mutation and other components we are going to discuss about is with respect to the migration, so it reduces the variation. Migration reduces the variation. especially when people are coming from the, like this, if you keep people are coming from the island to this, where variation is actually decreased. Okay, but overall genetic equilibrium gets affected. Okay. Let's see. Next, we'll talk about remaining factors. Yeah. So one other factor is non-random mating. Non-random mating. In the statement of Hardy-Weinberg equilibrium, we learn that in randomly mating populations, we say, correct? But we were also briefly talking about this, that random mating doesn't happen, at least in human populations, random mating doesn't happen. So we have some sort of preference in selecting the mate, correct? So there are different types of them. Here we are going to briefly talk about what is non-random mating. So Hardy-Weinberg equilibrium holds true only when there is no bias in choosing the partner. But in reality, that is not the case. So non-random mating happens. Random mating doesn't take place. So what takes place is non-random mating. So there is some amount of selection takes place in choosing the partner. Wherever you are not able to understand, do stop me. Okay, some of these are theoretical concepts. Okay, I'll try to simplify where possible and bring in examples. But wherever you are lost, do ask me. Okay, don't hesitate. So random mating means not selecting, not choosing the partners, marrying randomly. So there is equal chance of the genetic information being exchanged. Otherwise, because of the priority or because of the bias, what happens? Only certain amount of gene types will be favored over the other. Correct? The ideal population should be where randomly everyone, like a person from doesn't matter from one caste to another caste, one color to another color, is getting married to each other. Correct? If that is not happening, then you are creating bias in the population. So gene frequency can be affected if you are creating the bias. Okay, there are different types of them. We are going to talk about two of them. That is, one is assortative mating. There are two types of non-random mating types. So how does the selection work? One is called assortative mating. Selective mating basically. Assorting means, separating or selecting. So non-random mating based on phenotypic similarities, external similarities, phenotypes, appearance, looks. Okay. So when that happens, it is called assortative mating. So I don't know whether you study this in social anthropology as well. Humans pair bonds are established here based on what? Maybe religion. You have preference towards your own ethnic background, or maybe profession, maybe color. So color, caste, religion, ethnic background. So here some amount of selection is based on some factor like this. Okay, such mating is called assortative mating. Okay, so this takes place when sexually reproducing organisms tend to mate with individuals that are like them in some respect. So some amount of selection is happening here. When selection is happening, you refer it to as assortative mating or assortative pairing or bonding. Out of assortative mating, there are two types of them. One is positive assortative mating. Just briefly know, in case if they ask you a little bit more in-depth analysis on this, you add these key points. Otherwise, here you need to understand what is random mating and what does normally happen in populations, which is non-random mating. You should be able to explain in a few words what is non-random mating. So for that, I'm explaining it to you in the form of different types of them. One is assortative, another one is inbreeding. Inbreeding, we have a specific topic given in this chapter. in this unit. We'll focus more on it, but I'll give you definition here. So assortative mating is selection based. Sometimes it is positive, meaning people who have like features, they marry. That is positive assortative mating. Assortative mating. Or sometimes it is negative assortative mating, meaning dissimilar. People like the opposite ones, opposite colors or opposite types, opposite professions. Doctors may not like sometimes to marry doctors because they know how busy the life. So they may marry someone else from a different profession. They may be obsessed with what they do. This is one example, but sometimes, dissimilar. You also notice that there are cases where people with different characteristics tend to marry. In that case, it is called a negative assortative mating. And positive assortative mating is where like features, similar features. People with similar features get married. Okay. So what is the result of this then? If there's some amount of selection that is happening, what type of impact will that have on genetic equilibrium? So depending on whether it is positive or negative, either variation or genetic diversity will increase or decrease, correct? Genetic diversity will be affected. If people marry those that are like them, so particular type of genotype will increase. So bias will be created in the population. So genetic equilibrium will not be as expected by Aardvark because random mating is not happening. You are creating that bias through selection. So this would impact the variation. It would either reduce or increase depending on what exactly is happening, increase or decrease the variation of the population. Okay. Right. So non-random mating, specifically assortative mating, has this effect on the genetic equilibrium. Another one we need to know is inbreeding. Have you heard about inbreeding, the term inbreeding? Inbreeding. So inbreeding is breeding between close relatives. It is breeding between or mating between close relatives. Breeding between close relatives. Okay, so this is a very common practice, especially in South India, this is a common practice, correct? Or in some religions, it is a common practice. So we'll discuss more about inbreeding in the next part where we'll talk about consanguineous marriages. For now, just be familiar. This is a type of mating. Here we're talking about inbreeding. Marriage is happening within the families, within the relatives, within the blood relatives. So inbreeding happens there. What is the effect of inbreeding on gene frequency, genotypic frequency? We'll talk about it in a minute. Okay. Once I finish the factors, I'll get on to the details about consanguineous marriages that specifically given in the syllabus. So I'm going to focus more on inbreeding. Okay, on that. But for now, just be familiar that it is a marriage between or mating between close relatives. And when practiced repeatedly, it leads to an increase in the homozygosity of the population. How I'm going to explain that with example later. But yes, homozygosity of a population will increase. So thereby affecting the, so it is favoring one particular type of genotype, homozygous condition, either homozygous recessive or homozygous dominant. So thereby affecting the genetic equilibrium. Okay, we'll talk about the rest of the things in consanguineous marriages where we'll discuss about inbreeding, depression, and those components. So I don't know whether you need to know this much, but for all these things, there are mathematical ways to calculate the numbers. If you want to calculate the percent or amount of inbreeding that is happening, there is a mathematical term, a statistical term for that called coefficient of inbreeding. So it is calculating the probability of an individual being homozygous for certain locus, meaning at certain genes, like capital T, we talked about for testers, right? So if you want to calculate the probability of a person being homozygous for that particular region, so this is calculated using coefficient of inbreeding. Okay. So these numbers vary based on whether first cousin marriages, second cousin marriages. Okay, we'll talk about, I don't know whether you need to know so much about the numbers here, but just be familiar. There are ways to calculate. There are statistical or mathematical ways to calculate it. This is part of your handout. If you're interested, I can explain it to you further. But for now, just be familiar. If you want to learn more, read more about it, I'll explain you more. Otherwise, it may confuse you now. So, but be familiar that coefficient of inbreeding is a way to calculate the percentage or probability of carrying the dominant or recessive homozygous condition in a given individual or in a given population. Okay, I'll come to this genetic consequences later. Mutation we already finished talking about. So next we'll talk about natural selection. Natural selection, one of the factors. Natural selection. We finished mutation, gene flow, non-random mating. We need to talk about natural selection and genetic drift. So natural selection we already discussed in evolution, correct? Darwinism. Darwin has explained how natural selection act on certain things, right? Competition, survival of the fittest, and then those that are beneficial will be favored by natural selection, meaning they survive. Correct. So this survivability has a dependency on fitness of the population. We say that survival of the fittest. So what does it mean by fitness? In what terms they need to be fit? Do they need to be physically fit? Do they need to be mentally fit? Do they need to be fit in terms of their health? So what type of fitness do we talk about in population genetics or in evolution? Okay, so here it is reproductive fitness and survivability, that aspect of fitness we talk about. So this is a very fundamental concept in evolution, and it can be defined as, fitness can be defined as the ability to survive and reproduce. So fitness can be defined as ability to survive and reproduce. Okay.
Sir, one doubt.
Yeah.
Sir, natural selection can also work in favor of one who is not fit, right? In some chance.
Okay. Why do you think so? Why do you think it would favor someone who is not fit?
So let's say there's a population and they have some recessive trait in them.
Hmm.
Let's say they are dark skinned, let's say,
Hmm.
and that region has very high, high amount of insulation. So they will have better chance to, survive there.
Correct. Right. So that is what is fitness. So somehow that change of that character is helping them to survive better. So there's nothing called actually natural selection. Someone who is fit would survive. So we explain it using the concept of natural selection because they are fitter compared to the other characteristics or compared to the other individuals. So they survive. We refer to this concept as selection, natural selection. So you're implying that character is helping them to survive. How is it not, favoring the fitness here?
So I am saying if there is some white population which is more in number
Hmm.
that way, and these black ones are small in number. So even though they are less in number, but the recessive thing is helping them. So I'm saying
Correct.
that.
This is a very dynamic thing. Evolution is a very dynamic thing. Let's say, and natural selection can only act on what is available. There are always whites. They're only marrying whites. Natural selection has not too much to select. So when black people move to that place, when they have for some reason they have a fitness advantage, they would survive more than whites. So now natural selection has a new source to act on. So the larger population doesn't mean they are the most favored. They are the only available sources to the natural selection to act on. So whenever new change through mutation or through migration has been introduced into that population, they are favored, provided they actually found to be fit. Let's say people with Africa, they are, uh, resistant to malaria. Some of them have mutations to show resistance to malaria. Then what happens if malaria is more common in that area? They would survive. They are fitter. So more blacks will survive than the whites. But natural selection would act based on the fitness here. By numbers, it may sound that the favored ones are whites, but this is a dynamic scenario where black ones were never available. When they are available, they are fitter, they would survive over whites. Okay?
So essentially, we are saying that we are not seeing whatever fits becomes fitness.
Whatever increases the fitness of survivals and reproduction is favored. Okay? Right. So reproduction is a key here. To be evolutionarily fit, an organism must pass its genes on to the future generations. So why reproduction fitness is important? Because through reproduction only we are letting our genes to be transmitted to the next generation. This is a key event in evolution. And that is why that area is favored here. That area is actually focused here, has been in focus. So reproduction fitness would allow the transfer of genes to the next generation. So reproductive fitness is the key area or key aspect in evolution. The more fit individuals contribute, the more fit individuals contribute more to future generations than the less fit individuals. If people are sensitive to a disease, what happens? Their genes would make future generations more sensitive. So there's no point in a way. Actually, this is not happening because of a logic. This is how we are learning. This is naturally happening. People who are fit would survive or not, less fit would not survive. HIV, let's say HIV has become more common in Africa. People who have mutations to handle or show resistance to HIV would survive. Others anyway will die. We call this process natural selection. So fitter individuals would pass on their genes to future generations, making them better with modifications, continuous modification to it. Less fit individuals cannot handle this. They would die. So that is how fitness actually is a defining factor for evolution, defining factor for selecting something that is more useful, advantageous. Okay, so natural selection acts based on this fitness concept. But for that, we have to satisfy a couple of basic conditions, something that you already know. So what are the 3 basic conditions for natural selection to act? There must be inherited traits, meaning these characteristics have to pass on to generations. Otherwise, there is no point in selecting them or deselecting them, correct? So first of all, they act on something that is inheritable, moves from generation to generation. Second, there must be variation in these traits among members of the species. As you were saying, all of them were whites. What to choose? Now, with respect to the color, of course, there are many inherent differences in all those whites as well. But if you talk about color, all of them are whites. Nature has nothing to choose here. So there must be variation in these traits among these members. That is when selection happens. It's like, you're being selected for a particular sport, right? Your fitness is important. Let's say kabaddi or whatever sport. So there must be certain characteristics that your coach is looking for, a selecting committee is looking for. Let's say all of them are equal. What would the committee choose? There's nothing that committee can do there. Correct? So there need to be variation. Only when there is a variation, natural selection has a role to play. Otherwise, there is no role for natural selection. So there must be variation in these traits among members of the species. That is only when natural selection is required. And which is the case, which is always the case. We are frequently acquiring changes, mutations, recombinations always happening in us. And natural selection have a chance to choose. And next, these characteristics, not every characteristic is important. Do you think sometimes hair color, hair type makes a difference to survival? In humans, it may not. In birds, it may. Maybe it attracts the mates better, right? So these traits should affect fitness. Only those traits which affect the fitness are important. Those that are not, probably don't have. Like if you have appendix or not, doesn't matter. If you developed wisdom teeth or not, it doesn't matter because it is not affecting your fitness. So those characteristics which affect fitness are important for natural selection. Okay.
Sir, but it can also happen that, let's say, all of them are fit. The second point, if we don't have variation, but all of them are fit, so nature should select all of them, right? Since it doesn't have anything to select, but it has to select all of them.
Right. There's no selection happening. What does it mean by selecting all of them? Is any selection happening? Like in the example I gave you, the 10 people going for it, all of them are equal. There must be a mechanism to select them. There must be a criteria to select them. And that selection does not happen. In games, of course, if there's restricted numbers, only 10 can play with the 20 people, somehow you select it. Correct? But in nature, that's not the case. You don't have to kill few people. There's no need to kill people. So what happens? No selection is happening. The selection has not much role here. Correct? Selection only happens where people are not fit, they die. People who are fit, they are actually surviving. This itself is called selection. Indeed, as I've been telling you, there's nothing called selection happening physically here. People who are better, fit, they survive. Those who are not fit, they die. We call this process selection. But as it is, there's no one selecting or filtering out of for any reason. But as per the theory, we studied like this. Okay. The three types of selection, natural selections are there. I just want to briefly explain this to you. The three types of natural selections. I'm showing this to you in an image, which will be easier for you to follow. Three types of natural selection. This image nicely explains it. So natural selection can be directional selection, three types of them: directional, stabilizing, disruptive selection. Okay, I've given this briefly in the handout. Understand this from the image. Draw the image. If you want, draw the images. Directional selection, stabilizing selection, and disruptive selection. So natural selection, maybe of these three types. What are they? What is the difference between directional, stabilizing, and disruptive? So simply the concept is this. What is favored? What is favored? Sometimes it may go in one direction. One extreme trait may be favored. People, instead of dwarf people, maybe tall people are favored in a given population. So when it is happening in one direction towards one particular trait, you call it directional selection. So in this graph here, so let's say there are people who are short, who are medium, who are long or tall. So initially population was like this. On average, there were more medium people, more medium height people. But because the fitness, the advantage of being tall, if it is favored, what happens? Eventually, this population will shift from this initial type to this green color region you are seeing here. The people with more height or more taller people have survived because some reason they have advantage. In this particular example here, let's say there is a creature like this, which has a very short tail, versus something that has a big tail, which makes them to look like a snake and scare the predators. Is there an advantage if it is actually scaring the predator? So does it improve its chance of survival? If it has a long tail makes it look more scary. Would that be advantageous to it? It would, right? It would survive better. So what happens? This type of tail is preferred over short tail. So natural selection may favor this. In that case, you call this directional selection.
Sir, giraffe case.
Giraffe's case, long neck giraffes could be example for directional selection here. Okay, so long neck is an example, but towards one, basically, towards one extreme, towards one direction. Okay, such a selection is called directional selection. Next one is stabilizing selection. So what is stabilizing selection? Remember this, between all these three things, the difference is in what direction we are going. Here we are preferring one of the extremes. Here we are stabilizing it, meaning we are preferring something on average. So look at this. Here we are preferring something that is medium, neither short nor long. Something that is medium, something in between. And theoretical example for this is, look at this, something like a tiger. Let's say a tiger with short tail versus tiger with the long tail. Which is preferred? Neither of them. Why? Short-tailed ones will not provide enough balance because tail is normally balancing component of the body. If it is really short, it would not be able to use the tail for balance. So this doesn't provide much advantage. On the other end, the long tail, what happens with the long tail? It has to hold the tail and run. Our tail will be actually dragging on the ground. Will make it very difficult to run and catch its, the food. Correct? So neither long tail nor the short tail is beneficial. Then what should be the best case scenario for this? Tigers have, tails that look like this, intermediate tails. It satisfies the requirements of balancing and also doesn't cause a problem in chasing the animals that it needs to eat. So something intermediate is favored. In that case, it is called stabilizing. Something intermediate is being favored over the either extremes. Short and long, instead of short and long tails are long characteristics. An intermediate characteristic is favored. That is called stabilizing selection. Are you following this? The difference between direction and stabilizing selection?
So we always choose something in the middle for stabilizing.
Yes, it has to be intermediate. Yes, it has to be intermediate. I've given this short description in the material, and I've also provided some examples. We'll talk about those examples. Okay, once I'll explain the concept to you, I'll give you some human examples as well. Okay, so just understand the concept here. In the handout, I've given description that you can use in the exam. If they ask you about natural selection, talk about natural selection, its role, types of them for a short note. Okay, the last possibility, third possibility is, because initially we chose one of the extreme. Here we talked about something intermediate. Something called disruptive selection selects or favors both the extremes, two extremes. So short and long. So usually this is a population, right, with the intermediate features. But here in the disruptive selection, the result will be higher number of short and long people. Okay, so both extremes. So meaning both of these conditions provide some advantage, but intermediate one don't provide any advantage. Like in this particular case, as you see it, something like a squirrel, for example. Short tails help them keep predators from catching you on the ground. They're very short tail, otherwise, predator will catch that tail and pull it off. So tail can be problematic one for it. Correct? So short tails are beneficial because they can easily escape from the predators. Whereas long tails are very good in balancing on the trees. But medium tails neither help in escaping from the predators, nor do they help them balance well. So they don't have bigger advantage here. So either some of these in the population may keep the shorter tail or shortest tail here, or they have long tail. So if it is beneficial to be on the either side, you may find a few of the species, few of those members in the population will have shorter tail, few of them have the longer tail. If that happens, this is called disruptive selection. You are on the two extremes. Okay. I hope this concept of different types of natural selections are clear to you. Now let's look into human examples. Direction selection, stabilizing selection, disruptive selection. Okay. Yeah. So look at these examples. Directional selection. Okay, instead of shift in the variation in a consistent direction within a phenotypic range, we talked about that, going in one direction, preferring one of them. Now, for example, I think antibiotic resistance is not human relevant. So HIV resistance. HIV resistance in humans. Those people, like if there is an incidence of HIV is high in a given region, people with resistance to HIV will be favored, right? So the population will move towards the people who carry HIV resistance. So natural selection will move towards that direction. That is why it's called directional selection. Shift in the variation in a consistent direction towards one direction within that phenotypic range. Here, no resistance versus resistant. There's nothing called intermediate resistant here, right? So this is example for directional selection. Second, stabilizing selection. Something in between. Infant weight or infant size. So human birth weight. We'll talk about this in human growth and development, unit 10. Okay, but for now, just remember. So why can't we have, because there's a range for infants to be born. You know, you see it in the news. Children below 1.5 kgs are normally not preferred, meaning they won't survive well. Nowadays, because of technological intervention, even 500-gram infant can also be saved. But on average, the child should be, or infant should be 1.5 kgs approximately to survive. So there is a range. So the lower limit here is about 1.5 kg. Upper limit is about 3.5 kgs or maybe 4 kgs approximately, depending on the population. So there is a limitation of the infant size. Okay, so infant greater than or less than 3.4 kg have increased mortality. So the limitation is up to 1.5 kg, below 1.5 kg, survival rate of the babies is low. Above 3.5 kgs, babies cannot easily pass through the birth canal, because the problem here, the limitation is the birth canal, size of the birth canal. Humans in evolution tend to have a birth canal which restricts the baby size. So now we have to favor something in between, right? Not too low, not too high. So here we're talking about birth weight. Shouldn't be too small, shouldn't be too heavy. So you want something in between, because the otherwise death rate, mortality rate will increase. Smaller babies are more prone to problems. Heavier babies are more prone to delivery and also more prone to causing death to even mothers who are delivering. Of course, nowadays technical interventions again. C-section is more common. You can easily take the baby out by using an alternative channel to take the baby out. Okay, do you understand the example here? The relationship of this example to the stabilizing selection, something intermediate. Okay. Next. Example for disruptive selection, two extremes is gender, human genders. Male or a female. It does rarely happen. It does rarely happen that people who are in between is also possible. We'll talk about intersex, it is called. We'll talk about that in 9.4, chromosomal abnormalities. But it does rarely happen. But normally we have two extremes, one male, one female, like that. So this could be taken as an example for disruptive selection. People who have intermediate sex are not usually favored, but they can happen. They are rare, but they can happen. But nature doesn't seem to favor them. At least for our theoretical understanding, it doesn't favor. Okay, right. So this is briefly about natural selection. Natural selection, if it is present, do you think gene frequency or genotypic frequencies are affected? Does natural selection causes any change to the gene and genotypic frequencies? So from this, if you have to reword what we discussed, they ask you to explain, describe the role of natural selection on or effect of natural selection on Hardy-Weinberg law or Hardy-Weinberg equilibrium. What do you say?
Directional selection and majorly directional selection changes the variation. And stabilizing probably stabilizes variation.
Okay. Right. What about disruptive?
Again, this probably maybe stabilizes level.
All of them would. See, as far as you're adding a bias of selection, then you are changing the frequencies. Otherwise, there is a maintenance of equilibrium that is happening. Natural selection will affect it. You are favoring some or the other. Doesn't matter. One extreme, other extreme, two extremes, something in the middle. You are favoring something there. That favor will affect the genetic equilibrium. So genotypes like HIV resistance. So people with the HIV resistance, let's say people with the homozygous recessive condition for a particular gene, are favored. Small t numbers will increase. Capital T, small t, capital T, small t, capital T, capital T, capital T, small t genotypes are less favored. Assuming that recessive means you are knocking out the particular gene which is required for HIV to enter. HIV resistance normally happens through mutation in a gene which is a requirement for HIV to enter into our body, enter into our cells. So what happens? You mutate that gene. If that gene is and its product is not available, HIV cannot infect us. So meaning the nature is focusing or forcing the selection towards not having that functional gene. Correct? So it would impact the gene and genotypic frequencies. Because you're adding bias here. You're actually selecting something over the other. That is what, because natural selection acts on the fitter one. Fitter ones will be selected over the less fitter one. Then what happens? Frequencies change over a period of time. So natural selection does impact. So only in absence of natural selection does genetic equilibrium is maintained. So does genetic equilibrium is always maintained or anytime is it maintained? It never gets maintained. It's only theoretical cause. Okay, right. What else we need to learn? Genetic drift. Heterozygosity also we'll talk about somewhere else. We have balanced polymorphism. I'll talk about this later. Okay. Genetic heterozygosity. Selection coefficient. There's also mathematical terms for this, but I don't think this is important for you. So if you're interested, I'll let you read. Otherwise, you can ignore this component. Because I don't think they're asking this information. It's a little technical. Okay. But if you're interested, I think we are going to talk about it in other aspects. So there are mathematical ways to calculate the fitness. Genetic drift. This is a very important aspect. Can also be asked as a short note. Genetic drift. So what is genetic drift? Okay, so genetic drift. Drift means shift, change, moving away, drifting away. Drifting in the genetic gene pool or genetic information. Okay, so to explain it to you, first of all, let's look at the definition. It is the changing of the number of available alleles in a population by random chance. Okay, what does it mean? By chance. By chance, variation is affected. but it is more, this chance cannot cause huge impact on big populations, large populations. The impact will be high on small populations. So random or a chance event in small population results in genetic drift. What do I mean by that? I'll explain this with example. Okay, so random changes in the gene pool of a small population due to chance. Write down the statement. Changes in gene pool, genetic sequences of a small population due to random chance. So here we are learning about factors that influence Hardy-Weinberg equilibrium. So it should influence the or change the gene and genotypic frequencies. And here we are talking about random chances making that. Random chances affecting the genetic diversity. What are those random chances and why do they only affect small populations? What are those random chances and why do they only affect the small populations? Look at this example. So what do you notice here? In a small population of some bugs, there are two colors of bugs in that population. One is brown, another one is green. Someone accidentally stepped on them. So what happened? Most of the green ones are dead, leaving only one green one out. What will happen? What will happen in the future generations? Will there be more greens or more browns?
More browns.
More browns, right? The number is high. The mating between brown ones will be more likely. So there will be fewer green ones and more brown ones. So here it's a random chance, a random event. It could be a disaster. Okay, drought happened, or earthquake happened, fire accident happened. Whatever, there's a chance, a random chance. This is a random chance. And here, given that there is a difference in genetic diversity, because it might have taken enough time for them to give rise to these green ones, and accidentally they died. What happens? Brown ones will increase, and they have no choice here because there are only only a particular type of them. In a simplistic scenario here, genetic diversity is decreased because of the random event. Genetic diversity is decreased in that population. Okay, why is that more significant on small population versus large population? Here, the size of this population is very small. We have about 9 of them. Out of them, 3 greens, 6 browns. 2 greens have dead. What happens here? Given is a small population, the chance of them having a maximum effect is really, really high. Instead of this nine of them, if there are 100 of them, this event like this would not cause enough damage to them. I'll give you human examples to this, okay? For now, just focus on understanding this from this. I'll give you a well-known human example you can write as a case study. Okay, so it occurs, this is called a bottleneck effect. This is called a bottleneck effect. Bottleneck effect. This is also there in NCERT class 12th. They've explained briefly about Hardy-Weinberg equilibrium. Read that. Okay, class 12th. If you just type Hardy-Weinberg equilibrium, NCERT class 12, few pages are being dedicated for this topic. They briefly described about bottleneck effect, but this is a concept. Bottleneck effect means it's a random chance. It's one type of genetic drift. So what happens here? So in the bottle, why is it called bottleneck effect? If you see in the right side image, So this is the population. Several yellows, several blues are there. But when you actually try to pour out, what happens? They get stuck. Only few of them will come out. As you see it, those that are in this container could come out of the bottle. Rest of them died in a way. In a random chance, a random event, they died. Maybe flood happened. Only few of them could escape the flood. Rest of them died. So few of them will have now, compared to the genetic diversity of the initial population, the genetic diversity of this population is very, very less. Now they have to suffer in the process. They would only maintain, surviving individuals will have only fewer number. As you see, the initial population versus the final population, there were more yellows than the blues, but many yellows came out. So what happens? Sorry, many blue ones have come out. So more blue, the population became more bluer compared to the initial population, which was more yellowish. Yellow probably had fitness, higher fitness than blue ones, but because of the random event, genetic frequency has changed. So if that happens due to this random events, it is called a bottleneck effect. Bottleneck effect. Because of the bottleneck, only few of them survived. Out of them, those survived would replicate. But it changed the whole scenario of the population, whole composition of the population. Okay, are you able to follow this? This is called genetic drift, and bottleneck effect is one such type of genetic drift. So this is a very well-known factor. This is a very well-known example. Bottleneck effect. Excuse me.
Sir, if in this bottle case, what if most of them had come out and a few remained? Then it would have changed the scene.
Then you don't call it bottleneck, right? Bottleneck means where you are kind of choked. You could not come out. Only few of them could come out. Then it won't be bottleneck effect at all. So only those events where majority of the population has been affected, changing the genetic diversity, leaving out very few varieties. Okay, accidents. This could happen. There are many incidents where accidents have happened. People who are more prone, maybe females of them died over males. Okay, there are, if you say black and whites, blacks would survive if there is a higher temperatures in those regions over the whites. Or, if you talk about those industrialization concept, moths, dark moths over the fair ones, dark ones will survive. Okay, but genetic diversity has to be maintained, and if accidents happen on the small populations, they will be drastically affected. This is called bottleneck effect. And why does genetic diversity decrease here? So the outcome of genetic drift, bottleneck effect, is increase in genetic diversity or decrease in genetic diversity?
Decrease.
Decrease in genetic diversity. Don't they multiply later? They will
They
multiply.
do.
Okay, but don't genetic diversity will increase there?
Population is
Only one particular.
So what happens? Normally inbreeding happens there. Whatever the varieties that are present, they would undergo mating. So thereby resulting in more inbreeding. So diversity will decrease. It will not increase. Okay. So it will take time. Through mutations, again, diversity can be introduced, but it will take enough time. So overall, genetic diversity will decrease after a bottleneck event. Even though they multiply, I have given this in the handout. Out of necessity, populations that have experienced bottleneck increase their instances of inbreeding. They will result in more inbreeding to build the numbers to an acceptable level, so to increase the population. However, Inbreeding does not increase diversity. Instead, it decreases diversity. So because of this, what happens? After several generations, the gene frequency in that population will be significantly different from the gene frequency of the original population or initial population. Correct?
Sir, in certain situations, some factors have to be remain constant.
Can you repeat the question?
In certain situations, some factors have to be remain constant, like migration. If migration has forced it into that population, then genetic diversity will increase. If large migration takes place at that area.
There's a lot of disturbance in the background. So what I'm saying, there are instances where decrease in diversity is important. Am I getting it right?
Sir, if large migration takes place in that area,
Okay.
then it will the genetic diversity will increase.
Genetic diversity will increase. Okay. So, what is your question?
The question is, certain factors have to be remain constants.
Certain what?
Certain factors have to remain constant for this to work. But we're talking about small population, so
That is not applicable.
that is not applicable.
I'm having a hard difficulty in understanding your question here.
So Yash was suggesting that for this to work properly, he's suggesting that ideal conditions, certain ideal conditions must be met.
Correct. So we are only talking about situations where the case with a small population. So yes, that is not applicable here because large population it will not cause the effect. So random events would only impact, as I've been telling you, the small populations. If there is a big, huge population, this impact will not be seen. Bottleneck effect will not be seen on them. So this is applicable to small populations only. Okay. Right. The next second type of genetic drift is called founder's effect. So let's learn what is founder's effect. So founder's effect, as a term founders, come back to this. As a term founders here refers to, founder means someone who started the population in this case. Ancestors refers to the ancestors or the settlers, early settlers who colonized or who founded the new population in new areas. So founder's effect mainly refers to something like migrants, people who moved from one place to another place and established their own colonies. I'm not sure if you learned about Amish populations in who moved to North America from Germany. So some populations in olden days they have actually moved and established in new areas. But what they did, they actually restricted them to themselves. They marry within them. So a lot of inbreeding happened in them. Thereby genetic diversity is eventually lost. So small population, few people. In one example, I'm going to tell you there 200 people moved to North America. Okay, an example here, Amish community. Amish people in USA, Pennsylvania, for example, there are about 200 individuals. About 200 individuals migrated from Germany. So very small people, small number of people have migrated to North America. This became a very good case study because such rarely these type of things happen. But for genetics, because we cannot control humans, but already existing conditions can be taken, examples can be taken to understand this. So what happened? These people tend to show a lot of genetic problems. They had a few recessive alleles in them, but because of inbreeding, they kept spreading those problems in their community because of the marriage within them. Recessive alleles came together, causing more and more homozygous condition with the effect. So they carry some unusual concentrations of genetic mutations, causing a number of otherwise rare inherited problems became more common. Dwarfism became more common. multiple fingers became more common. Some of them are not deadly to them, but the rare conditions have become more common there because there are only small number, small founding population, okay, and they were inbreeding, resulting in lesser and lesser diversity. That is called founder's effect. That is called founder's effect. Okay, you see the community here. So this community, people actually have, people are short. They have very short limbs. For example, dwarfism is very common, and some features like this, as you see. Do you notice their fingers? So this is called Ellis-Van Creveld syndrome. Ellis-Van Creveld syndrome. They have recessive alleles causing this disease. As they married, homozygous condition became common there. And these people have extra fingers and very short limbs. Compared to the whole world, very majority of the examples are from Amish community of this disease. Because some of them, out of those 200, some of them carried this allele. They kept marrying within the same family. So what happened? That allele got distributed among them. And when it appeared in the homozygous condition, you could see this. But you can ask if it is lethal, why do they survive? It doesn't look to be lethal. It is not lethal, but it causes effect. The small fingers, it would not kill them, but it would impact their lifestyles. So they could still survive and spread this problem. So rare genetic problems become more and more common theme in the founding communities like this. So there were 200 founders and now there are 250,000 people and they spread around US, Canada, in many states. Okay, so this is called founder's effect. Okay, so like that, either founder's effect or bottleneck effect would would influence the gene and genotypic frequencies. It would reduce the genetic diversity. Okay. Genetic drift.
Can you show the definition for founders effect?
Sure. And this is part of the handout that I'm going to send you today. So founder's effect happens in the small populations. Okay, who live isolated from others. Okay, with more and more inbreeding happening there. Such an effect is called founder's effect because they start really small and what results in there is inbreeding. So reduce genetic diversity. Small population to begin with, isolation, inbreeding, reduce genetic diversity. These 4 terms you can use to explain it. Okay. Right. What else are we left with? Migration we discussed. Isolation. So sometimes you also notice that isolation can also impact it. Isolation means like either this is overlaps with what we already discussed. So some sort of separation, like either because of religious, geographical, like island, mountains, glaciers that we were talking about. So may separate people like tribals or other people are separated and they get genetically isolated. When they get genetically isolated, again lesser and lesser diversity, more inbreeding. Okay, by chance, if few alleles are not inherited to the other generations, it will even extinct. Those alleles will be gone. Okay, so these things can be commonly seen in the founding populations we just talked about. Gene frequencies in small isolated populations do not reflect those of the larger founding population from which they were derived. So what happens if the population size increases, the diversity increases, the effect cannot be seen, but otherwise the small populations would have, isolated populations would have the same effect that we just discussed in genetic drift. Okay, this can happen within the population as well. We talked about in the speciation. Religious isolates, people who are religiously bound, where marriages outside the religion is forbidden. So what happens? They also notice founder effect. They don't have to be living separately, but these people isolated themselves within the same community by restricting them to marry others. So what happens? Genetic diversity also decrease. There are Pakistani communities in UK where a study called, we'll talk about that study, born in Bradford study, where we'll notice that those people have actually restricted to themselves, resulting in lesser genetic diversity, higher incidence of genetic issues. Okay, so we'll talk about that next. That actually takes us to the next topic in unit 9.3, because so far we discussed about the various factors: mutation, isolation, migration, selection. We also briefly talked about inbreeding and genetic drift. So these are the factors given in the syllabus affecting the Hardy-Weinberg equilibrium. So you need to know what is Hardy-Weinberg equilibrium, what is its importance, what are the assumptions on which Hardy-Weinberg equilibrium work, and what factors influence Hardy-Weinberg equilibrium. Okay, are you comfortable with this content here? Okay, so we'll continue. Yes.
In isolation, we also observe the founder's effect, right?
In isolation, we also observe the founder's effect, but large populations will not have that effect, but small populations, those who are isolated within the small numbers, would have this effect. So it is an overlapping theme basically. Isolation is not separate individual concept. It overlaps with what we learned in genetic drift. As it is given in the syllabus, I wanted to briefly touch upon isolation. Isolation means they're physically separated or religiously separated or for any other reason separated from the big population, thereby reducing the diversity.
So can we quote Sentinelese tribe in this from Andaman?
You can, yes. Those type of people are they founders? I have not learned about them, but all those tribes might be physically separated, correct? So
Yes,
they can
sir.
be taken as example for genetic drift or isolation. Okay.
Yes, sir.
So any tribes, any small populations where they restrict to themselves could be taken as examples. So you can pick up several of them from socio-cultural anthropology part. Okay. So next, we'll touch upon the related concept here, that is consanguineous marriage. The next topic in unit 9.3 is consanguineous and non-consanguineous mating, leaving out genetic load, genetic effect of consanguineous and cousin marriages. Okay, I think partly you touch upon some of these aspects in socio-cultural anthropology, but here I'm going to briefly touch on what are consanguineous marriages and what is the genetic impact of that and why does it happen. Okay, so this could be taken as part of assortative mating that we discussed. Assortative mating, we talked about that, where physical traits are cultural, based on cultural conventions. People select the mate, people select the partner, correct? So one such thing is consanguineous marriages. So consanguineous marriages refers to mating between the blood relatives or close relatives. In Latin, con refers to common, sanguineous refers to blood. So blood related marriages are marriages between close relatives. By definition, so some general definition is this: a union between two individuals who are related as second cousins or even closer than second cousins. Or a bit more technically, you can say people with inbreeding coefficient equal or higher than 0.0156. Inbreeding coefficient. Homozygosity. Remember, I was briefly talking to you about inbreeding coefficient. It is the probability of identifying that or finding homozygous condition for a given gene. Okay, this can be calculated using inbreeding coefficient. Shweta, you are trying to write something on this.
Sorry, sir.
No.
By mistake.
No issue.
Sir, can one look at the genes and find the coefficient?
Can one?
How do we find the coefficient? Is it required for the syllabus?
No, it is not required for the syllabus. It is just, as I was telling you, this is one kind of, if you want to get into the depth of how is that calculated, because I'm going to use some numbers. So I want to have a general idea about what is inbreeding coefficient. Because from a study, I'm going to show you the how are they related religion-wise, how much of consanguineous mating is seen in different religions. So for that, I wanted to introduce the concept of inbreeding coefficient briefly, but I don't think this will be asked in the exam directly, but you can make a mention like inbreeding coefficient, where there is a probability of finding homozygous condition for a given gene, can be a value one can use to calculate it technically. Okay, so as per a 2001 study given by Bittles is equal to or higher than 0.0156. So higher the homozygous condition being seen, higher the connection between those numbers. Okay, some communities like Arabs and in South India, uncle niece marriages that are common. So there you will find highest inbreeding coefficients. So supposed to be at least 0.01, right? It will be 10 times more than this. In some communities, you will find 0.125 inbreeding coefficient values, 10 times more than the defined value. Examples: Arabs and uncle and niece marriages in South India. Okay. So I'd like to give you brief statistics on this. This is to strengthen your answers. If they're asking you to write about consanguineous marriages, cite these type of things. Some of these are old studies. Okay. So if you're interested, I can also provide you some case studies on South India. I remember there are some articles on this. Okay, but they're very specific communities. But I would like to provide you some generic idea here. So from a 1999 study, types of cousin marriages permitted worldwide, if you see. So first cousin marriages, 39% first cousin marriages are allowed in the world. 39%. And second cousin marriages are 34%. This data is taken from over 1200 societies in the world. So first cousin marriages are 39% common as per 2000, so 20 years old study. And the second cousin marriage is 34% common. And remaining is neither first nor second. Okay, either you pick up this information or I'm going to give you religious-based marriages as well. Okay. I'm sure you probably have studied this chart in your socio-anthropology class, right? Cross cousin marriages, parallel cousin marriages. So these types you study. You can also talk about them. Types of cousin marriages also. As per the demand of the question, you can briefly talk about it. So father, sister, father's brother, mother, sister, mother's brother. Okay. So here this chart is important. So consanguineous marriages and the percentage. So first look at these numbers. Hindu marriages. If you see 10.6% consanguineous marriages happen in Hindu marriages from a study. And where is the highest? Muslim religions normally have 23 higher percentage of consanguineous marriages. 23% approximately. These numbers may vary from data to data, but higher incidence. Over 20% of consanguineous marriages percentage in Muslim communities. And next, actually interestingly, Buddhist communities. 17% in Buddhist communities. So after Muslims and Buddhists comes the Hindus and Christians. About over 10% in both Hindus and Christians. Christians and Hindus. Okay, below 10%, you will see 6 very small, Jains 4.3%.
Sir, who is conducted this study?
Sorry, I forgot to mention that. Let me share that article with you. Okay, I don't think I have made a note for this. Okay, yes, let me check that and give you the reference for that. Okay, I forgot to add the reference here. I have the article with me, so I'll send you that article or the link to that article. Okay, but this looks to me probably from the old study like this. Okay, I'll post that in the Telegram channel. Next question is, why do they marry? Why consanguineous marriages? Are there any benefits of consanguineous marriages?
Purity.
Okay, one is the concept of eugenics. So purity, they want to maintain the same characteristics. And they may feel that they are more purer than the others. Correct? So these type of concepts. Olden days, when the eugenics concept was happening, they preferred something like this, marriages within the families.
Trust is also one.
Hmm?
Trust.
Trust, exactly. Trust. More family support, less financial pressure. If they have to pay more dowry, they prefer to marry within the families to reduce that. And to gain the sense of security and comfort. Also, they want to have these marriages happen within the families, within the known communities, within the blood relatives even. And also because of the family support, it could reduce the divorce rates as well. There's many family members are involved in this. And also to keep the properties to themselves. Correct? If they don't want, because if they feel that someone else will exploit the properties in the name of the marriage, they would marry within the families to maintain. So there are many social reasons why it happens. have a lot of articles on this, but I will provide you a brief summary on this. If you're interested, I can share some of these articles with you. I think there's one article, Consanguinity and its relevance to clinical genetics, published in 2001. So some of these can be used as case studies. Social and economic factors associated with consanguinity. So you can extract this information. And the clinical effects. Okay, I'm going to describe that to you. If you're interested, I can provide you this article. Okay. So what are the effects of consanguineous matings or consanguineous marriages? Because if you see the syllabus, there's, first of all, we have to explain what is consanguineous marriage and what are the different types of consanguineous marriages, and then some examples of them. And then we need to describe about the effects of the genetic effect of consanguineous marriages or cousin marriages. So mainly it will introduce the rare alleles in homozygous condition. So it would increase the homozygosity. The main impact of consanguineous marriages is if there are rare alleles, as we have seen in the founder's effect, the rare alleles, those rare alleles will be maintained in the population. Because those rare alleles should go away, right? If they're causing any harm or if they're reducing the fitness of the population. But if the more and more inbreeding happening, because consanguineous marriage also involves inbreeding, right? They're marrying within the families, within the same groups, who are related to each other. So what happens because of the rare alleles are maintained? Some of these recessive alleles are maybe not found in other populations, but because these groups are actually marrying within them, these are maintained. Okay. So because of that, what happens? One, it decreases the genetic diversity. Genetic diversity would decrease, homozygosity will increase. With respect to that particular locus, homozygosity will increase, decreasing genetic diversity and decreasing heterozygosity. So heterozygous conditions will decrease, homozygous condition will increase, genetic diversity will decrease. So if homozygosity increases, what happens? Autosomal recessive disorders. Autosomal recessive disorders, because sex chromosome recessive disorders are only common in females. So mostly what gets affected here is autosomal diseases. Because only condition where sex chromosomes in recessive condition can be harmful is XX condition. So most common condition would be autosomal condition. Doesn't matter, chromosome 1, chromosome 2, all other autosomal chromosomes. If two people got married who carry same recessive allele, which is rare, meaning if a person who married to other family, unrelated family, then very less chance to find same mutation in the other family as well, right? So what happens? So because of the consanguineous marriages, because of the inbreeding here, so two defective allele carrying individuals can come together. These genes can come together, thereby resulting in the rare autosomal recessive disorder. So this disease burden will increase because of that, because of the increased probability of homozygous recessive genotypes. Okay, person A married to person B. Both A and B are related through their ancestors. So what happens? A can carry same mutation, B can carry same mutations. But if A married to someone else, to C, B married to someone else, D, they may not carry same rare mutation. The chance is very, very low. But if they marry within the family, both recessive alleles can come together when they marry, resulting in that rare disorder to show up. Okay, because of that, overall incidence of genetic diseases will become high. How high? Sometimes you may ask, is it really lethal to marry within the families? It may not be. Okay, it will increase the genetic disease burden rate. By what percentage? The estimations show that about 2 to 3% higher than the normal incidence. So it is not too high indeed. Okay. But even if it happens, even if there is a rare allele that is present, then it would result in such a problem. Otherwise, less than 5% chance of increased genetic disease burden in that family. So that goes case by case. Okay. In 1 million population, 1 billion population, you will find enough examples where people have higher impact on their group. But otherwise, it increases by about 2 to 3%. Okay. So for this, I'm going to send you, there is a newspaper article, I think 2 articles I collected on this, referring to this project called Born in Bradford project. It's called BIB project or Born in Bradford project, conducted in England. So Pakistani Muslims living in UK and England, study has been done on them, and the genetic disease burden rate is high in them because they prefer to marry within them and resulting in less and less diversity, more homozygosity. So Born in Bradford. I'll share that article with you. Read that article. If you have any doubts, ask me the questions on them. In next class, I'll explain that to you. Similarly, the studies done in Norway double the risk of congenital defects in consanguineous marriages. Okay, so they have clearly shown that it would increase the risk of congenital defects, birth defects through consanguineous marriages. Okay, next, possible increase in Down syndrome. We'll learn this in 9.4. Down syndrome would not inherit. Normally, it is not a, it is a chromosomal problem, but a parent who had Down syndrome will not inherit that to next generation. So it doesn't run in families, but in risk factor for that may increase. So the reason why Down syndrome happened, we'll learn that the mechanism. That problem may persist in the families, if not exact disease. Okay, you'll understand it later when once I explain you the Down syndrome in unit 9.4. The defects of consanguineous marriage and genetic drift are often operating together in small populations. So what happens in small population? Genetic drift had happened, small population will marry within them. So consanguineous marriages are common. So thereby both genetic drift and consanguineous marriage like inbreeding would affect it. Okay, so increased disease burden, decreased diversity, increase the chances of problems like chromosome abnormalities like Down syndrome are the problems that you notice because of consanguineous marriages. Okay. So you can strengthen these answers later because there are a few more things that we have to learn. Okay. So you cannot write a complete answer on something like this. To strengthen this, we have to learn few more concepts, and those concepts will be discussed under 9.4 and 9.1. Okay. So you will eventually develop the concept related to this better. But is it clear what is consanguineous marriages? What are the different types of consanguineous marriages? So for that, you can use that something like this that you would study: cross cousin, parallel cousin marriages, first cousin, second cousin marriages. And you can show information like this religiously, how common this is in different religions and what studies are showing. And what is the impact or benefits of consanguineous marriages and what are the negative impact of consanguineous marriages. Okay.
Sir,
Yes.
they are autosomal recessive disorders. That means they don't inherit.
The recessive disorders means the diseases only show when both defective alleles are present. And each allele comes from one parent, right? So person A and person B both carry that allele, negative allele, recessive allele, but it is masked by the dominant allele. Like the case with hemophilia, like the case with sickle cell anemia. What happens? Only when these two defective alleles come together in a child, that is when they show up. That is when it causes lethality. So they become more and more common because related people are marrying. They're carrying recessive alleles. So homozygous condition becomes possible because of this. Okay. So we studied this in 9.2 in Mendelian genetics. We talked about those dominant and recessive alleles. Okay. So this is an extension to that. Right. So we'll stop the class here. So today we discussed about the remaining factors affecting Hardy-Weinberg equilibrium, consanguineous and non-consanguineous marriages. We are left with one, genetic load, and we are left with genetic polymorphism. Those two concepts I'm going to discuss in next class. So I'm planning to take a class tomorrow as well. I'll confirm that tomorrow morning. So Sunday I will not be able to take class, but I'm planning to take class tomorrow. Okay. So I'll post the class schedule in the morning. Okay, so the next class will finish 9.3. We're going to discuss genetic load briefly. We already talked about basics related to genetic load. I need to explain what is genetic load if short note comes and examples related to that, types, mainly types related to that. And then we'll talk about genetic polymorphism. This question is coming in the exam usually, especially balanced genetic polymorphism, transient genetic polymorphism. These concepts are being asked in the exam. Okay, I'm going to explain that. Otherwise, from this unit, the most important one being the Hardy-Weinberg equilibrium and factors affecting Hardy-Weinberg equilibrium. That is the most important question from this unit, and it is usually repeated. Okay, right.
Sir, when if this question is asked in the exam, depending on, say, for short notes or a long notes. So for short notes, we'll just explain what Hardy-Weinberg equilibrium is, and then we'll also mention factors affecting it.
Yes, so you briefly talk about depending on the demand of the question. Let me pull the So let me check that question. Depending on the demand of the question, I think I have a video I made after 2018 UPSC exam. If you go to YouTube channel of Feynman, I've answered this question. I've discussed this question there. Have a look at it. I've given key points for you to make a note of. That is really useful to you. So otherwise, okay. So 2017, they gave simply Hardy-Weinberg law. So you give the statement, you explain the assumptions, give an example. Okay, they only gave Hardy-Weinberg law. There you don't have to discuss this 10-marker question. You just mention the factors because anyway in the statement factors are given. You only have to expand what are those evolutionary factors. Okay, if 10-marker question is asked like that, you talk about that. Second, 2011, a question was asked where write short note on conditions necessary for the operation of Hardy-Weinberg law. So there you have to focus on assumptions under which Hardy-Weinberg equilibrium work. So the question demand is different. And one question, 2015 question, what they asked here is discuss the factors affecting gene frequencies among human populations. Factors affecting gene frequencies among human populations. So what do? You talk about the factors we discussed today in the second part of the last class. And another question that came is on what are the genetic effects of, you have this PDF with you, right? For this questions bank. The question bank PDF shared with you, am I right?
Yes, sir.
So if you check that, I think this version had page 63, page 62 and 63 questions are given from that. You have a look at it and see if you can answer those questions. What are the genetic effects of consanguinity? Give examples. So there, as I was telling you, you have to give examples from the other studies, hemophilia, sickle cell anemia, or Amish communities example you can give. Okay. So our Born in Bradford project example you can give. Okay, right. So Yash, I'm going to actually, to the question that you asked, I'm probably will answer that question in the Telegram group to you, or as I see that it is an interest to others as well, remind me when I'm beginning the next class, I'll briefly talk to you about that. Okay, so synthetic theory and Darwinism comparison you asked me, right?
Yes, sir.
Okay, right guys, so otherwise we're going to stop the class here.
Okay, sir. Thank you.
Bye everyone. Yeah.